Good evening, everybody. Welcome. It's a real thrill to hosting a new inaugural lecture. Tonight it's Kirsty's turn, and I am thrilled to say she's joining us actually from the Department of Infection and Immunology at St George's, but she isn't always in the country, as we will go on to learn. For those of you that are on successfully, that's great. If people text you or contact you at the start of the lecture and for some reason they're unable to find the link, I'd be grateful if you could tell them to turn to the event page on the first and opening page of the St George's University website. So we have access to the link through the button on the event page at the front page of St George's own webcast. So I'm the principal of the university, and I have to say that Kirsty in many ways has been a record breaker, and that includes tonight because more people have registered for this event, and I think a very diverse and international audience are tuning in to hear. But I hope also a spattering or indeed a large collection of family and friends who don't normally hear about your scientific work and background. And I think it's always lovely for those events as well to incorporate your family and friends who may learn things about you that they didn't previously know. In terms of background, some of you may know that Kirsty didn't start in medicine. She had a background in economics and then worked for 10 years for non-governmental organisations, but then was a success story of the graduate entry programme here with us. She went on after graduation to specialise in paediatric infectious diseases and immunology, and as part of her academic training also rotated around to Imperial College London. She's always been interested in age-related immune responses to infectious diseases, and in particular has focused on group B Streptococcus. Now, my background is originally in that of obstetrics, and I can promise you the devastation and harm and problems caused by this pesky little bug are awful. And I was so delighted to hear this focus of the work that Kirsty's doing, because it really does shatter lives. Her interests are very broad. They relate to the basic pathophysiology, the immunity, the epidemiology, the public health interventions, with the overall aim of reducing the impact of this infection. She collaborates so widely and is also part of the WHO task force to defeat meningitis and developing a vaccine for the condition. As I said before, she travels extremely widely. I dare say that has been somewhat curtailed by the pandemic, but it's lovely to have this opportunity to hear a resume of your work to date. We're not in the room together, but at the end, there are some people with you there. That's absolutely fantastic to see. Paul Heath is going to give the vote of thanks. We don't do questions as part of an inaugural lecture, but you will see that the chat function is open if you want to say, make a comment there. But if you ask a pesky little difficult number, Kirsty will have to take it offline and answer it because it won't be answered this evening. So thank you very much indeed for joining us this evening, and thank you very much, Kirsty. We look forward very much to what you've got to say.
Thank you very much, Jenny, and good afternoon, good evening, everybody. I will be talking in the next few minutes about group B Streptococcus and why, after 50 years, we still don't have a vaccine. So first of all, what is group B Streptococcus? Well, it's commonly found in the gut and the lower vaginal tract of most humans, but it's still the leading cause of neonatal infections, both early onset in the first week of life and late onset up to the first 3 months in many countries. And if a baby gets GBS disease, the case fatality rate is quite high. The global incidence is approximately 0.5 per thousand live births, but it's double that in sub-Saharan Africa at over 1 per 1,000 live births. And it also causes disease in pregnant women, it causes stillbirths, and it causes preterm birth. So clearly there's a huge need for prevention of this devastating bacterium. But compared to other forms of research, group B streptococcal research has tailed behind. Although it was first discovered in the 1800s, and case reports in the 50s and 60s showed that GBS was a cause of neonatal infection. It wasn't until I was born in the 1970s that data started to come from the US highlighting that GBS was the leading cause of meningitis in the first 2 months of life, with about half of babies that got GBS disease dying, and of those that survived, up to half had severe neurological sequelae. And similar findings were found in the UK, where over half of babies that got GBS sepsis died. So you would think that with this impetus and this knowledge that a vaccine would be soon forthcoming, knowing what we know about other vaccines that are in development. But again, compared to other vaccine preventable diseases such as pertussis, the amount of work that has gone on around GBS disease is much less, and this is a sign of both funding and focus, I think. But even with maternal pertussis vaccination, which came about in 2011 following the pertussis epidemic, the amount of research hasn't really matched that of other diseases. So clearly a focus is still much needed on GBS in particular. But it all started as with Carol Baker, and here on the right side is Carol in the lab, who was the first to demonstrate in the 1970s that antibody transferred from mother to child could protect from disease. And she published this wonderful paper in the mid-70s with 2 different strains of GBS, which showed that babies with low concentrations of antibodies were much more likely to have disease than babies with higher antibodies passed from their mother. And this is really the concept of maternal vaccination, not just for GBS, but she demonstrated that this was possible for all potential bugs that happen in the first few months of life. And hot on the tails of this was the first generation of the GBS vaccine, which was a native polysaccharide vaccine. And this is a study from 1978 from Carol's group, which although it demonstrated that there was some immune response, for some of the serotypes, such as serotype 1A of GBS, the responses were very variable and quite low. And this is probably because of the reliance on T-cell independent B-cell activation. So there was no memory to the infection created by B cells. And in this study, the titers plateaued even after boosting the second dose. So it was really back to the drawing board at this point. However, 10 years later, the vaccine had been reformulated and the first trials happened in pregnant women. And the vaccine was now conjugated with a tetanus toxoid, which made it much more immunogenic. And this study again from Carol and her team showed that the vaccine was safe and immunogenic, not just at delivery, but in the baby at birth and up to 2 months of life. So you would have thought that by the time I went to university that there would be a vaccine. However, although this data was published, the vaccine was still not forthcoming. And during all of this time, I had finished school and started on a European-funded international programme, which saw me travel to London, to Madrid, to Reims, and to Reutlingen. And the major thing that happened during my studies was that in 1989, the Berlin Wall came down. And really for this European cohort of students, me included, this was a real sign of a reunification of Europe, and it was a real desire by all of us to help rebuild the ex-Eastern Bloc and to work together to build a larger and more cohesive unit, and really taught us about thinking together. And I graduated from this degree in 1993, and this being Germany, we all put on our best jeans for the celebration in the car park. However, I graduated in Germany at a time when unemployment was rising for the first time since the 1950s. And so, as Jenny mentioned, I had a series of interesting jobs, including a short stint as mayor hostess with Lufthansa City Line, an even shorter time with Helles & Moritz before moving into the IT sector and consulting, with digital equipment in India and with Sega in Japan. And then further consulting with the food industry with Phileas Fogg, where I was involved in the first Virgin Global Challenger that didn't go around the world in 80 days and sadly landed in the Atlantic. And then on to Barilla Pasta and development of a Barilla Pasta business in the UK. And finally into NGO work with Oxfam. And through all of these different consulting roles, I learned a lot about international working and international project development, which would stand me in good stead for what was to come. So in 1996, after all of this, the CDC published the first recommendations for the prevention of group B streptococcal disease. And this was a real breakthrough for GBS prevention and control. When I was practicing this talk with my husband yesterday, he pointed out that actually the most important thing in 1996 was that we got married. But for me, this is up there in the top 3. So shortly after 1996, the second generation glycoconjugate vaccines were developed. And these, as I said, were conjugated to a tetanus toxoid, which made them much more immunogenic. And you could see that compared to the plain polysaccharide on the left, the white spots have far higher antibody concentrations. In the middle, you can see that there's a good correlation between the amount of antibody that was generated and the ability to kill GBS in vitro. And compared to non-vaccination, people were boosted with higher antibody concentrations. And this is because the conjugation aids memory B cell induction and allows class switching, which makes the vaccine much more immunogenic. So by 2000, you would have thought, again, that these vaccines were now ready to go. However, at the turn of the century, after I had had my 2 children, Megan and Crystal, we were 30 years and counting since Carol Baker published her first reports of GBS disease. And yet still, at the time of both my pregnancies, I wasn't offered any vaccine or prevention to GBS because that wasn't available in the countries where I had my children. So in 2000, I took the leap from working in NGOs to graduate entry medicine, having seen an advert in The Guardian for graduates with no qualifications in the sciences to join the medical school for graduate entry medicine. And this is a 4-year scheme that's based on the US and the Dutch schemes, where you don't have to have science background to be able to study medicine. And that we were the second year of the JET course, and here you can see many of my colleagues all have gone on to do bright and wonderful things, both at home and internationally. And because the JET course was so self-directed, I had lots of opportunities to do other things. And my friend and colleague Yolan Knight here in the white shirt, who was a postdoctoral neurologist before starting the JET course, made the rash decision of taking me into her laboratory for 18 months of headache research. She helped me get my personal license, and I then spent time using immunohistochemistry to test nociception following stimulation in neck pain as a cause for headache. And through this, I learned 3 things. The first was that I really hate animal research. Immunohistochemistry is really hard, but also that I enjoyed lab work. And this set me up for postgraduate academic foundation training with Ian McPhee. And again, Ian, for reasons best known to himself, decided it would be a good idea to give me a lab project growing Jurkat cells and testing toxicity with tacrolimus on them. And I can guarantee you this isn't a picture of my cells because I managed to kill them before we even got pictures and certainly before the challenge of tacrolimus. But I still enjoyed lab work at this point, and that led me to academic paediatric training. However, once I'd started paediatric training, I began to miss the field, and it was quite clear that I wanted to go back and combine paediatrics with work in resource poor settings. And Mary Cummins and Camilla Kingdon took the brave decision of allowing me to leave paediatric training and train to be a GP so that I could gain the obstetric and anaesthetic skills that I needed to go and work as a medical doctor in Babisa Hospital in Matuba in KwaZulu-Natal, and with the Africa Centre, which is now the Africa Health Research Institute. And there I worked with Ruth Bland and Marie-Louise Newell, developing a Wellcome Trust proposal looking at HIV-exposed and infected infants and their neurodevelopmental outcomes. However, all did not go to plan, and Wellcome Trust said no. So at this point, I returned to paediatrics and met these three professors in a time when PIDRG celebrated its parties on Mike's boat. The Vaccine Institute was a small room behind the blood bank, and we used to meet in Tatty's office of Pinkney Ward at St George's. But whilst I was working with them, I became involved with the cabin study, and together we published some papers on infection in children with and without cancer, and also, for me, most importantly, looking at the effect of antibiotics in resource-poor settings. And this was the first time, really, that I thought I really wanted to combine infectious diseases with working overseas and trying to work to reduce the global burden of mortality in neonates and infants. And this is where my journey with GBS started. So Paul, who does occasionally smile, did what every self-respecting PI does and asked me to write a review article. But most importantly, he introduced me to Beata Kampmann, who also asked me to write a review article. And these two really have been my supervisors, mentors and friends ever since I first met them back in 2011, and are part of the reason that I'm here today. And with these two leaders in science, I then went on to develop my second Wellcome Trust proposal, which was to go and work with Beata in the Gambia at the MRC unit. And we developed a proposal to look at the seroepidemiology of GBS in the Gambia to help with vaccine trials, and set that up and submitted that to Wellcome. However, all didn't go to plan, and Wellcome said no again. So I returned to paediatric training and had a third go. And finally in 2014, 2 years after the first submission, I finally made it to work with Beata's team in the Gambia. And I spent 2 years developing field and laboratory work together with these two beautiful teams of people, many of whom I'm still in contact with. And we had some great times both in the Gambia, at international conferences, and in the lab to develop the serocorrelates work. And during that time, I also met Chrissy Jones, who suggested that it might be an idea to expand the serocorrelates work that I was doing in the Gambia in a population of HIV-exposed uninfected infants and HIV-infected infants from her PhD. And together we were able to demonstrate that although HIV-infected women had lower concentrations of antibody, the subclasses of the antibody that they passed were actually in proportion. And this is really important because it suggests that we might need different vaccine schedules in the context of HIV infection. But most importantly, it suggests that if you can boost the antibody in women with HIV infection, then you can protect their infants just as well as in a woman who's not exposed to HIV. And in my PhD, I met Andy Gorringe and Steve Taylor, who have supported me in all of the lab work that I have ever done since starting my PhD. And as I realised when I was putting this together, I've actually been with the group so long that we've been through 3 iterations of Public Health England. When I started, we were the Health Protection Agency. During my PhD, we became Public Health England. And recently we've now become the UK Health Security Agency. And I continue to work with these two lovely people and the great teams. And they and the group have taught me everything that I know about flow cytometry. Again, not one of my plots, but I occasionally got mine to look like this as well. And Andy suggested during my PhD that it might be a good idea to learn about opsophagocytosis assays in an industrial setting. And so I had a short trip to Siena, to Novartis, where I was able to work with Monica Fabrini and Imam Margiti Ross, to learn the assays that Novartis were using in their vaccine trials for GBS. And not only was this a great experience learning how lab works within industry, but it was also wonderful because I got to be in Siena in the autumn when it was still beautifully sunny. So the upshot of my PhD was that we were able to pinpoint an amount of antibody above which babies were not colonised with GBS for the main serotypes in the Gambia, which were serotype 2, 3 and 5. And you can see on the top are the observed thresholds that we saw, and then at the bottom is the calculated thresholds. And this is the first time that we were able to link GBS colonisation with antibody in blood. And importantly, for a population like infants in the Gambia, where breastfeeding provides a huge source of protection, we were also able to show, using the complement deposition assays that were developed at Public Health England, that antibody in breast milk was associated both with clearance of colonisation in the baby and also with an absence of colonisation. And this is really important when we're thinking about vaccines, because if you can boost antibodies in milk, this might be an additional role of protection. So whilst all of this was going on, Carol Baker and her group published the first data which suggested that we could pinpoint an amount of antibodies, so a serocorrelate of protection, above which an infant wouldn't get GBS disease. And for serotypes 1A, 3 and 5, the calculated amount of antibody was 1 microgram per mil or more for a 70% risk reduction. And that was 90% in serotype 1A on the left and 3 in the middle, and 70% for serotype 5. And Shabir Mahdi's group went on to demonstrate that a serocorrelate of protection was also possible in South Africa. And hot on the heels of this, in 2016, data from the GSK vaccine trial was published. And this vaccine was a third generation vaccine which applied scientific advances to GBS vaccine design. So using reverse vaccinology to identify key vaccine targets, they developed a trivalent vaccine, 1A, 1B and 3 serotypes. And in this dose ranging study in South Africa, led by Shabir, and in Malawi, led by Rob Haydeman, pregnant women without HIV were dosed with 3 different doses of the GBS vaccine. And you can see here that all of the coloured bars show a good response irrespective of the dose, and the antibodies persisted for up to 1 year post-delivery. So surely by 2016, we should be ready for a vaccine. However, as I said at the beginning, although GBS is an unmet and important medical need, the low incidence of GBS makes vaccine efficacy studies long and complex. And Shabir's group demonstrated that with a disease rate of 0.1% and 75% vaccine efficacy, we would need approximately 90,000 women recruited and vaccinated to get 43 cases of GBS disease per protocol. And this is obviously huge logistically, very costly and almost impossible in most countries to achieve. And so we needed an alternative. And together with Beata and Paul and several others, we went on to publish the data on serocorrelates and protection that was around, and made this a call to arms for alternatives to licensure. And then in 2017, we had a breakthrough for potential alternatives to licensure. First, we presented to the Medicines Health Research Agency and the European Medicines Agency in 2017, and then to the Food and Drug Administration in the US in 2018. And both looked favourably on serocorrelates of protection. And here is the group celebrating at the plaque commemorating Alexander Fleming's lab at Imperial College back in 2017 when we started this work. And really, once we had this pathway to licensure, several studies were then able to start. So in the UK, together with Paul and Public Health England, a serocorrelate study of 150,000 women. In the USA, a similar study using dried blood spots. And in South Africa, a similar study, which is about to finish. And also in Uganda. So 4 seroepidemiological studies in different countries, together with 2 vaccine studies in South Africa, Uganda and the UK, all using the same assays that are developed by the team here at St George's, to be able to make the translation between the different studies possible for the first. And in order to develop these standardised assays, first we had to develop a protocol, and to do that, during my postdoctoral time, I was sent to Moon Nam's lab at the University of Alabama in Birmingham to learn his miniaturization technique. And here I am with Rob, who's his laboratory manager, learning the miniaturization technique. And on the right again, it's not my plate, although eventually, through Rob's persistence, my plates ended up looking a little bit like this. But they're very hard. And then came my UKRI Future Leaders Fellowship. And this gave me the opportunity to work with a whole fantasy football team of experts in neonatal research. In addition to Beata and Paul and Andy, I was able to work with experts in laboratory assay development, David and Moon, with experts in clinical trials, Shabir and Claire, Philippa and Annette, with experts in epidemiology, Stephanie and Joy, and of course with Carol, who's been a mentor for some time now, and with Jane Plum, who is the vocal advocate for Group B Streptococcus from the patient perspective. And for all the football fans in the room, I realise that there are more than 11 people on this chart, but I wanted to make sure I included everybody. So what about the vaccines? Well, Pfizer have now published their phase 1/2 trial of a hexavalent capsular polysaccharide conjugate vaccine. And here you can see for 3 serotypes, 1A, 1B and 3, that there is excellent immunogenicity at all doses with and without adjuvant compared to placebo in the blue line. And this is a study of non-HIV infected healthy adults and shows immunogenicity up to 6 months. And last year, Minovax also published their data on their protein-based vaccine. And again, this dose-ranging study shows in non-HIV infected healthy adults, shows immunogenicity up to 12 months, and there were no safety signals in any of the Minovax studies. So what about colonisation? So Sharon Hillier in 2019 also published data from Carol's tetanus toxoid vaccination study demonstrating efficacy against serotype 3 of 36% in time to colonisation post-vaccine, which suggests that the vaccine might also have an additional role in preventing colonisation during that key period of birth when GBS can be transferred from mother to child, on top of being able to prevent invasive disease. And the WHO and international momentum is also now behind the vaccine. So the WHO published preferred product characteristics and the roadmap in 2017, and in November of last year published the full vaccine value assessment. And I've been privileged to work with the group that have developed these guidelines. But all of these serocorrelate studies have occurred either in the USA, in Europe or Sub-Saharan Africa. But if we're to get a truly global vaccine, we need serocorrelate studies elsewhere. And it's really wonderful that Tatiana in Brazil and Meiwa in Japan are now starting their own serocorrelate studies in their respective countries, because this will really make the case for vaccine efficacy when the vaccine is finally in phase 4 trials. And there are also 2 new vaccine manufacturers specifically targeted for low and middle income countries. The Biovac Institute in South Africa is developing a multivalent vaccine, and PATH, together with Inventprice, is also developing a multivalent vaccine. And I'm really looking forward to seeing the data from their first trials. So during all of this time, having set up the lab with Tom Hall and colleagues, we've moved from 1 small and very cramped lab doing lots of work to 4 large labs with staff working on a range of different projects. And it's important to remember that I wouldn't be here today without the people working in these labs. So at the end of last year at the ISAD conference, We were able to demonstrate for the standardized assay that there is really good correlation between all of the laboratories taking part for the assay, which means that it's really robust and it's ready for use in the seroepidemiological studies that are currently ongoing. So what are we doing now in the lab? Well, Tom, together with Olwyn, Laxmi and now Sasmita, are working to develop assays for human infection models, and they've successfully managed to develop assays that measure antibodies in serum, in vaginal secretions, nasal secretions, and soon breast milk. And here you can see the difference between colonised and non-colonised non-pregnant women, and that you can also see that there is a difference between colonised and non-colonised in serum, but not in vaginal and nasal secretions. They also highlighted important similarities, but also differences in terms of the serotype distribution between the UK and Uganda, with serotype 5 being much more prevalent in Uganda. And this will be important when considering vaccines and that use serotypes. Also in the UK, together with Paul, we're running a serocorrelative protection study, which is led by Costas, Madeline and Topper. And Costas has demonstrated that we can pinpoint antibody decline for 5 of the major serotypes, which will be important if we're thinking about these studies. And the correlation between antibody birth compared to 4 weeks and 8 weeks is really good. At 12 weeks, slightly less good, but very small numbers. But this is great because this means that we might not need to collect cord blood on all mothers and babies, and we might be able to get blood from babies with disease and matched controls, which will be far less logistically challenging. Susie and Camille and Simon are developing gut organoids to investigate late onset GBS disease. And this is a study that collects human neonatal gut tissue at different gestational ages to develop a gut in a petri dish to measure immunity, because there's so much that we don't know about why babies get late onset GBS disease. And this is a picture of the first crick that was grown from the organoids. And I'm really looking forward to seeing the results. And as with many things, Susie and our first ever academic clinical fellow, Sarah, have also been working on a COVID study looking at seroepidemiology of COVID antibodies in pregnant women. And this is them presenting at one of the St George's research days. And of course, this goes hand in hand with the work that we're doing in Uganda on seroepidemiology and surveillance, led by Dr. Mary Chohere and microbiologist Lauren Hookham. And here you can see the Dr. Mary talking to the small army of the village health team that go door to door to identify babies that have risk factors for sepsis or signs of sepsis, to make sure that they get to hospital for us to investigate and treat them. And through this study, they demonstrated that GBS incidence in Uganda is approximately 1 per 1,000 live births, which is similar to other sub-Saharan African countries, and that GBS was associated with 3% of all stillbirths in the cohort. Also in the lab, Cato has been looking at the genomic sequencing of the isolates from my PhD to try and refine vaccine coverage for the Gambia. And Hamza in Uganda will be taking over Kay's code to do the same in Uganda. And Kay is about to defend her PhD thesis. So watch this space for what's coming next. And also in Uganda, we're preparing for these clinical trials that I talked about. And Dr. Roberts, Dr. Hannah and Rakam are developing a pregnancy register at Kawempe National Referral Hospital, which has approximately 21,000 deliveries every year. And again, we have a huge array of midwives and nursing staff that help us with the pregnancy register. And we're currently running the first of these 2 trials, the Minivax phase 2 trial, which has just recruited its 50th patient today. And obviously, hand in hand with all of this is working with the community to get them to understand about vaccines and vaccine safety. And in the top left, you can see Hannah filming one of the many videos that we have made that are being shown in antenatal clinics across Kampala about GBS, sepsis and recognition. And finally, we also do a large amount of functional immunity work in the collaboration with Steve Coase at the MRC-UBRI in Entebbe. And we have an array of guests that come to visit us for training in functional assays together with the team in the UK. And if anybody is fortunate enough to come and visit us, the Ugandan Virus Institute actually owns the Zika forest, which is where Zika was first isolated. And so, coming to the end of the 50-year cycle, where are we in terms of the current vaccine development? Well, finally, we seem to have made considerable progress in terms of defining regulatory and policy pathways for maternal immunisation and its acceptability during pregnancy. And the vaccine has been evaluated in over 3,000 pregnant women. Overall, different vaccines have been evaluated in over 3,000 pregnant women. But there's still much more to do. We need to benchmark vaccine-induced immunity against natural immune responses associated with a risk reduction in infants. We need to evaluate the immune response in a variety of settings. And we need to implement vaccination through phase 4 studies, which should include not just early and late onset disease, but also safety stillbirths and premature births because of the large burden associated with GBS. But finally, it feels like we're nearly there. And so with that, I'd like to thank the team in Uganda, without which none of the Ugandan work would have been possible, and the team in the UK, without which none of the UK work would have been possible. And finally, my family, thank you all very much for allowing me to travel the world in pursuit of a bacteria. Thank you very much.
Well, thank you, Kirsty. There is thunderous applause in the background there. Alas, we can't hear it, but it's undoubtedly there. And there are more than 100 people who have viewed this. Were they all with you in the lecture theatre? As I say, it would be thunderous. So it falls to me to give the vote of thanks, and I'm delighted and honoured to do so. Professor Hyam and other distinguished members of the audience, thank you, Kirsty, for your fascinating and entertaining inaugural lecture, which was truly professorial. If I may, I would just like to emphasize the significance of Kirsty's work in maternal immunization, in infant immunity, and in perinatal infection, and particularly in the context of her work in Africa. Her leadership in assay standardization for assessing candidate Group B strep vaccines, which she only touched on, but there were so many things that she was able to tell us today. But her leadership in that particular aspect is absolutely critical for the eventual approval and rollout of Group B strep vaccines, and they will have a major impact on children's health globally. This development will in turn facilitate the development, approval and implementation of other maternal vaccines against important pathogens. But more than that, she has now established a unique maternal vaccination platform in a low-income country in Africa. And this will lead to greater equity of access to vaccine research and vaccine deployment for these populations in sub-Saharan Africa, where most of the disease burden lies. The pitifully slow rollout of COVID vaccines globally has reminded us how important it is to address this issue, which is what Kirsty is doing. Now, I also want to say I struggle to manage my own work in one country, yet Kirsty is simultaneously coordinating clinical and laboratory teams, as well as administrative and College of Pediatrics responsibilities in 2 countries, which are 4,000 miles apart. And she does all of this with a great sense of humor, but also with attention and care to the individual needs of all members of her teams, and also the careful nurturing of young investigators, as I think she's shown us. So let me again thank Kirsty for her excellent presentation and congratulate her on this well-deserved professorship. We'll certainly find a time to celebrate her achievements, and unlike Boris, she will not need to bring her own bottle. Thank you, Kirsty, and thank you to everyone for attending Kirsty's lecture tonight. I'm pleased now to draw this session to a close. Thank you, and have a nice evening.
Thank you.